So let's now finish our discussion about protein folding. And so I want to start out this last section of this lecture with just a bit of a reminder of some of the big picture things we've been discussing about how proteins fold, right? How we think this process basically happens, right? So here's our sort of cautiously written down, folding pathway for a protein, right? Sort of your general protein. The first thing we know that happens when you roll a protein off the ribosome, or it's been unfolded because I'm an experimentalist and I did that on purpose, or maybe a stress condition, something blew the polypeptide up. The first thing that happens, right, as we discussed last time, is formation of the hydrophobic collapse of those hydrophobic amino acids that want to be in the core of the protein, and formation of local secondary structure, right? And we discussed the idea that in some cases, one or the other of those things is going to happen first, followed by subsequent folding events. In many cases, these two things happen at the same time, so-called nucleation condensation model. And these events tend to be very, very fast. This hydrophobic collapse and local secondary structure formation can happen on the microsecond time scale, about as fast as a polypeptide pieces can diffuse in aqueous solution, up to the millisecond time scale. So this is a very fast set of events. Second, depending on the protein, it can populate intermediates, or so-called partially folded states. These are typically compact. They're usually, though, more expanded than the native state. They can have significant secondary-like structure. In some cases, almost all the secondary structure of a protein can be folded by the time you populate one of these folding intermediate states. But they generally have no stable secondary structure. They're almost always still highly hydrated, right, or significantly hydrated, much more so than the native state. They're also quite varied, right? They have, there's no single consistent protein folding intermediate like the molten globule that people thought was going to be the Rosetta Stone for how proteins fold. Many proteins don't populate intermediates at all. Others populate things that look a lot like molten globules. Others populate other weird types of folding intermediates. So they're not always there, right? Okay. Then finally, the final arrangements that can take things out of either out of a well-populated intermediate, or sometimes you don't have a well-populated intermediate. You have the unfolded state, but you have some other events that really limit slow folding, so that you don't do this on fully to the native state on these very fast timescales. And that's fairly typical. And that's sort of the final docking of that internal structure of the polypeptide, the final packing in, right? Squeezing out of all that internal water, and really getting the internal structure of that protein snuggled down in exactly the right place, pinning down the last little pieces that are the most entropically difficult to get into place. Also, proline isomerizations. We talked about cis-trans proline isomerizations. The equilibrium constant between that cis and trans configuration is so low that in the unfolded state, there's lots of prolines that'll flip into the cis configuration, but they have to be trans for the polypeptide to be going the right way to fold. And that isomerization event can be slow on the minutes timescale. Right, so this can dramatically limit the ability of a protein to get down here into the native state. And then, of course, there's some other final events that are, in many cases, essential for a well-folded protein, like disulfide bond formation, right? Okay, so that's sort of the sort of grand overview of this sort of how do proteins fold in the sort of the simplest sort of high point way. It's significantly more complicated than that when you dig into detail. It's a fascinating problem, both biophysically and biologically. And the reason it gets fascinating biologically is because the folding landscape picture that I gave you before was for well-behaved, fairly simple proteins that fold well. Well, lots of proteins in a living cell, many proteins in excess of, say, 20 or 30 kilodaltons in mass that have relatively complex folding topologies. And what I mean by folding topology is bits of polypeptide that are remote in primary sequence that have to make intimate contact in the folded state. Proteins like this often have really complicated folding landscapes. They're not smooth. They're not even with like a single intermediate. They have all kinds of local free energy minima down here, holes, valleys, mountains, all kinds of stuff. And so you can imagine proteins up here trying to go down here, trying to get to this native state, right? Collapsing down and getting stuck in one of these local holes, right? the landscape, or so, excuse me, the pathway view of this is something called an off-pathway intermediate, a protein that would come up here, folding from the unfolded state, collapsing down, and then by virtue of some set of contacts that are reasonably stabilizing but wrong, the protein goes sideways, right, and ends up in a state that has no direct access to the native state. So it has to be disrupted, or it has to rearrange to get back on the productive pathway. Now, the reason this becomes such a problem for so many cellular proteins is this slows their folding. In some cases, if you have a well-populated off-pathway intermediate, it's incompletely folded, and either a protein that's folding slowly through these steps or an off-pathway state is going to expose contact surface, excuse me, interaction surfaces that should be satisfied in the folded protein. But now they're hanging out exposed. And that leads to all kinds of bad problems. Aggregation, where these proteins start to stick to themselves and they just turn into a big inert goo, is one of them. Sometimes they can interact with other things, membranes, other biomolecules in ways they're not supposed to be interacting. And all of this stuff, right? This is a huge problem. This is like a massively deadly, deadly, deadly problem. And the interesting thing is, this is such a deep problem in basic protein biology that evolution solved this problem very, very early on. And it didn't solve it simply by optimizing all proteins so that this never happens, right? Large numbers of proteins, many, many essential proteins, suffer from problems like this in a really big way. And evolution's solution to that problem was to create a set of cellular machines known, of course, as molecular chaperones. And molecular chaperones spend their entire lives shepherding proteins through the various stages of that protein's life, all the way from its synthesis off the ribosome to its folding to an active state, to assembly and disassembly of molecular complexes, transport across membranes, all sorts of things. Molecular chaperones are involved in virtually every aspect of significant protein dynamics. Not all, but a lot of the basic dynamics of protein assembly and disassembly and quality control that goes on in a living cell involves a molecular chaperone. And there are half a dozen, 8 or so different major families of molecular chaperones. I'm just going to point out a few features of them, but they are all basically designed as kinetic editors. And you get right down to it. They're all designed to inhibit that process of these off-pathway intermediate formation or loss of a protein down one of these local so-called kinetic traps. Okay, so here's an example of one that I happen to like a lot, known as GROEL. This is a member of the so-called HSP60 family, or also known as the chaperonins. It's one of the very first molecular chaperones recognized. And these guys are constructed of 14, in the case of the bacterial variant, 14 52 kilodalton monomers that are arranged in 2 7-membered rings, right? So here's one ring here, here's the other ring here. And the striking thing of these molecules is this open solvent-filled cavity, right? And that open cavity is what will recognize and bind a non-native protein that is getting into trouble when this protein is going to aggregate. And then as a result of binding both ATP and a little partner protein, a smaller molecular chaperone by the name of GROES, this is this little lid-like guy here. This protein forms a little folding nanocage, which you can see right here. So when ATP binds down here, GROES binds up here. The polypeptide that was captured here gets enclosed inside this little nanocage. And that does 2 things. One, it keeps it from interacting with anything else getting in trouble. So it forms this little infinite dilution box. But the interaction of the protein, the folding intermediate, with the chaperonin also changes the rules of the game. What we've discovered over the last 10 or 15 years is that folding of a protein inside this nanocage is not the same as folding of that protein in free solution. It folds better and it folds more efficiently, right? And that's just really kind of cool, right? Here's another example, right, of a protein that functions as a kinetic editor. You have all these proteins that, and this is super important in eukaryotic cells for proteins that are going to be secreted, right? Many secreted proteins have disulfide bonds, but you can have proteins with those disulfide bonds that, depending upon how they first collapse, the bond goes in the wrong way, right? Here's an example of one, right? Say we've got 4 cysteines, and they've paired up in the 1, 2, 4, 3, but that's the wrong pair, right? But that's a covalent bond. If it stays like this, the protein is dead, right? Protein disulfide isomerases have these paired cysteine groups in their active site that will attack. One of them is reduced. It will attack that disulfide bond to form a mixed disulfide, which releases and pumps electrons into one of the cysteine group, one of the sulfurs, which allows it now to go and attack the neighboring group. The polypeptide can rearrange. The other release group will then come back to now form this sort of like 1, 4, 2, 3, kind of like my little example protein before, and then release the PDI. These guys can also be used to put in disulfide bonds to begin with. So this is another example of a completely different class of molecular chaperone. Should point out that the protein disulfide isomerases are an example of a chaperone that don't directly use metabolic energy, whereas the chaperonins, these guys do, right? These guys burn ATP directly to drive this conformational cycle that folds proteins. PDIs require reducing equivalents, but they don't actually use ATP directly, right? Okay, so that, avoiding these rugged landscapes, navigating proteins around these nasty little traps is all about what molecular chaperones do when they assist in protein folding, right? So that kind of brings me back to this question I left you guys with at the beginning of our discussion of protein structure, protein folding, which is how on earth do we understand this phenomenon of prions, right? This idea that you've got non-nucleic acid transmission of replicated information, phenotypic information, and it's only based on a protein. How on earth is that possible? Well, what it turns out to be the case is the nature of the fold or refolding of that protein that in its normal condition, so-called PRP, it's innocuous and it does what it's supposed to do when in neuronal guidance or biology or whatever it's doing. But when this protein refolds, so to the so-called PRP-SC form, that's the prion form, PRPC is the form which is normal. Notice this protein is almost completely alpha helical. But this protein, when it because of mutations or because of stress or other sets of conditions, it changes its conformational state, it refolds. And this same amino acid sequence is compatible with a fold that's very enriched in beta sheet. And this is the kicker. This version of this protein has unsatisfied hydrogen bonding off the edges of that beta sheet and an interaction surface here on the top of that sheet that really, really likes to stack with another protein that looks like this, right? And then form beta sheet, a continuing beta sheet, right? As you stack those guys up next to each other to form something known as a cross beta superhelix or amyloid, right? And it's the conversion of this material into this material and the recruitment of more of this into this that makes these amyloid fibrils and that leads to diseases like prion diseases, Alzheimer's disease, Huntington's disease, and other classes of amyloid-like or amyloid-based protein misfolding diseases. Okay, we'll stop there.